All three dogs will have Play-Doh like in their ears, up their nose.
It'll be fine. All right, so the answer is A. Good job. Looks like we're at 100% there of the 15 people who... Do y'all not have clickers somewhere? Somebody missing some?
We have like 7
All right, we're not actually doing that yet. That's supposed to be after this section. All right, so bear with me for all this stuff because it's a little bit complicated, very zoomed in. Before we get into it, though, let's kind of remind ourselves of what's going on at a neuron that nothing's happening at. So first thing to remember, we've got like the dendrite end, right? These dendrites sticking out. Then you've got the cell body, the nucleus, and then a long axon, and then the axon terminal down here. So dendrite. And if you notice, all the neurons y'all drew were better than this one. Yeah, I know. All right. So the inside of the dendrite, is it more negative or positive than outside the cell? Okay, so we got some negative charges in there. It's positive outside. What about the cell body? Negative or positive inside? A little less confident out there. It's also negative. So negative, negative, negative. Positive outside. What about the axon itself over here? It's what? It is. What about the axon terminal? It's also negative. So when a neuron's not sending signals, the whole thing is negative. Just want to make sure we're on the same page there. And the way they send signals is by making it positive. So you let positive ions in, and that's going to trigger more positive ions to come in all the way from the dendrite all the way to the end.
Is a neuron ever not sending signals?
Yeah, all the time. So for example, you've got your pain neurons in your fingers. Is anybody in pain in their fingers right now?
Well, I guess I won't mean the pain ones, but I mean like every part of your body's doing something all
right.
the time, isn't it? Not necessarily pain or...
Mm-hmm. And as you'll see kind of later, you actually have specific neurons for all kinds of sensations. So you've got pressure neurons sending information about pressure. You've got neurons telling you that you're holding your arm like this. You've got neurons telling you whether you're in pain, neurons telling you the temperature around you. But each one of those is a separate neuron.
Okay. So even sensory neurons are even divided further into what types?
Exactly, yeah. Definitely. So yeah, you could have, some neurons in this spot that are sending signals about temperature, but the ones next to it are pain neurons, so they're not sending any signals.
Nervous system is simple.
Yeah, it's totally simple. All right, so first off, a lot of times the book just says action potential, but a lot of people don't know what that phrase actually means. So all an action potential is the event that changes the inside of the neuron to become positive and then puts it back to being negative. That's what an action potential is. So it goes from unactivated neuron, activated neuron, back to unactivated. So it goes from negative to positive inside and then back to negative. And another vocab word that you'll see a lot, depolarization. Does anybody know what that is?
When it's becoming positive.
Okay, so that would be when the inside of the cell becomes positive. And what do you think repolarization is? It's back to negative. So an action potential consists of depolarization followed by repolarization. So as you saw last chapter, or last lecture, the dendrite is the receiving end of the neuron. So it receives all the signals. Now, depending on what kind of neuron we're talking about, it could be a neurotransmitter is binding to some receptor out here and letting positive ions in. So neurotransmitter comes by. You can imagine it's this pin cap. Hops over, binds there, and it's going to let these plus ions through. So remember we did all those channels? What kind of channel is that? If something binds to it and it opens. So it's the ligand-gated or ligand-gated. So that'd be one kind of neuron. Neurotransmitter comes from another neuron, binds to it, opens the gate, positive ions come in. Or maybe it's a sensory neuron, like pressure. So we start out, negative, and then that neuron senses that there's pressure on your fingers, which opens up mechanically gated channels. So it opens the channel there, and now those positive ions, sodium usually, can come in. Same thing with the temperature neuron. That's mechanically gated too. It'll let your positive ions come in there. So that would be the start of our action potential of the dendrite. Put those positive ions back. Obviously, that's not the whole story. That's the start. So you've got those ligand-gated or mechanically gated channels that open in response to neurotransmitters or, mechanical stimulation. That's the start. And then what would come next is the voltage-gated channels open. So you've got voltage-gated channels that open when the inside of the cell gets a certain amount positive, which, if you can tell from our graph, what do you think the number, what kind of membrane potential has them open? So that's about minus 55 there, which means the inside of the cell got 15 more millivolts positive. And so step one, right, we let in a few positive ions. And then step two is those initial ions change the voltage, make it more positive. And now these other voltage-gated things start opening. So they start opening, and more positive ions can come in. And so if you see on that graph now, it very rapidly shot up to plus 30. So it went from minus 55 all the way to plus 30. So it became very, very positive inside from all those sodium ions pouring in.
So when the inside of the cell becomes more positive, that kickstarts the voltage ones to let more in as well?
Right. So it's like a two-step process. Step one is let in some sodium ions. And then once it gets positive enough, then those other channels open, the voltage-gated ones.
So the voltage-gated channels won't ever work without either of the channels or a ligand-gated channel.
Unless you get electrocuted or something. Um, so, and if you notice, what do you think this little one here is? This little pink thing, the little hump. Would be the ligand-gated ones. Let me kind of go back here. So for that little bump there, that's if the signal's not strong enough to get to minus 55. So then the voltage-gated ones never open. So to take an example, your pain neurons, for example, maybe they're sensing a tiny bit of pain at the moment, but it's not quite enough pain to actually create a signal. Like only a few receptors open, and they're letting, I don't know, 1 sodium ion in. It's not enough to get enough positive to get to minus 55. So step 2, step 3, none of that ever happens. Because the sodium ion gets pumped back out. Or, for example, the pressure one. There's probably some pressure on our fingers all the time, but it's not enough pressure to open enough channels to let enough ions in to get to minus 55. So that's what's called like threshold.
So I keep seeing the minus 70, Michael. Is that a standard number, at least that they've found inside of our
Yes, yeah. So your neurons, nearly all of them are about minus 70. That's just how they were evolved to be. And you remember last time they maintained that by pumping ions out and keeping the phosphates and proteins inside.
So if you have someone that has like nerve damage and they can't feel pressure, pain, temperature, anything with that, it basically changes their threshold.
If you're talking about, was that from like trauma or something, or something they were born with?
Trauma.
So trauma is usually like damage more in like the spinal cord or something. So like basically breaking the axon somewhere. Now there are genetic conditions where these things would be broken, like one of those ion channels or something, perhaps. Or even it's somewhat common to be like just born without one kind of neuron. Like maybe, because there's people who like don't sense any pain, right? Or at least they don't sense certain types of pain. That would be, yeah. Like break her leg or like.
She stuck her head, and how they figured out it happened to her was she stuck her head on the hot stove and made scream.
Oh my goodness.
Burn in her hand, that's how it happened when she was little.
That's pretty scary.
So like people that like are born blind so they're born without that type of
I mean, there's all kinds of reasons to be blind, but
it affects, but ultimately what happens is that it's affecting that.
could affect like the vision neurons, or it could have been like some mutation in the brain, so you're like sending signals, but they're not interpreted right. So anyways, so what does reaching membrane potential threshold mean? Discuss in your group. What does that word mean? There you go. So it's that minus 55. Or you could say letting in enough sodium ions to open the voltage-gated channels. Be another way to say it. So as we kind of discussed, once you get to threshold, that minus 55, or enough positive ions come in from this first part at the dendrite, those voltage-gated sodium channels open, sodium ions move in, inside becomes positive. see enough, right? Of course, right now we're still at the dendrite, right? So we've got a bunch of positive ions in there. We reached threshold. We opened up the voltage-gated ones, but we haven't sent any signals down here yet. Now step 3 is getting the signal from the dendrite to the axon terminal way down there. And this is pretty simple because all throughout the cell body, the axon terminal, you have more of those voltage-gated sodium channels. So if we've let in all these positive ions here, at this, like right here, do you think we've reached threshold? This little spot? There's definitely positive ions there, right? So that means that sodium channel is going to open, and then this positive ion comes in. And then there's a channel next to it. Do you think that one's reached threshold? It's gotten pretty positive, so it opens, lets in a positive ion. What about right next to that? Are you going to make me go along the whole neuron and do this?
No.
So that one opens and goes through. And that just continues all through the cell body, you know, opening up the channels because it gets positive. And it's almost like positive reinforcement or something, right? Because the one right next to it lets in an ion, which then opens the one next to it, which lets in an ion. So that's how it travels down. And this is the fastest way you could transmit signals through your body. Way faster than like putting a hormone into the blood and letting it get pumped around or something. Because as you know, how fast does electricity travel? Pretty fast, right? Faster than sound. Faster than water, for sure, which is what the blood would be. So this electrical signal goes down, opening up, and then when you get to the axon terminal, then you'd most likely be releasing a neurotransmitter from the end. So NT released. We'll talk about that last step a little later. So there's the propagation, just what I was diagramming and talking about. It's just the adjacent regions reaching threshold, which means voltage-gated channels open, sodium ions move in, and then it just continues itself on down. All right, so obviously something has to happen to return the neuron back to its regular state, right? So it's going to be ready to send more signals. And so the way that works is voltage-gated potassium channels open. And they're going to let, because if you remember, this is a little confusing, but remember potassium was higher inside the cell. It's one of the things it maintained. Remember that sodium-potassium pump? So potassium ions are very high in there. So we'll put them in green maybe. So the potassium ions are going to start flowing out through their own channels. So potassium. And then as the membrane potential gets closer to negative, the sodium-potassium pump starts pumping the sodiums out and the potassium back in.
What, is it just because it's positive? Is that what signals that to start?
Yeah, so you have voltage-gated potassium channels that open when the membrane potential is positive. So that like plus 30 that we got to. So as soon as you get positive, they open, potassium starts leaving, which is making it more negative, right? Because positive charges are leaving. And then you're also doing the sodium-potassium pump at the same time. because it's basically always active. And I know it's confusing. To me, it would make more sense just send the sodium back out directly, right? But instead, it's more of a two-step process. So let the potassium go out. And the main reason for that is the potassium's at such a different concentration that it goes out very, very, very quickly. So you can get yourself back to negative super fast. And then you can fix your sodium-potassium levels a little bit slower with that pump. It's going to pump the sodium out and the potassium in. Is that on there? So let's watch this all happen a little bit. And then I'll have you correct a paragraph. Let's see.
Must be rapidly conducted over long distances.
Hold on, I'll restart it.
In order for the nervous system to communicate with other cells, action potentials must be rapidly conducted over long distances. The processes that allow the conduction or propagation of an action potential along the entire membrane of an axon are not very different from those that generate one at the trigger zone. A positive feedback cycle is set in motion when a graded potential reaches threshold and initiates the action potential at the trigger zone of the axon.
So trigger zone's basically the dendrite they're talking about. I mean, it's basically the dendrite. But yeah, I guess right at where the axon is, technically. But for our purposes, it's pretty similar.
Depolarization of the first segment of membrane progresses as the sodium gates continue to open and lasts until the charge is completely reversed. When the action potential reaches its peak, the positive nature of the membrane causes potentials to spread to the adjacent segment of the membrane.
So if you notice our little diagram here, ECF, that's like outside the cell. And then down here, cytosol, that'd be inside the cell. And then in green is the sodium channel, and yellow is the potassium. And so at the start, what was moving? So the sodium channels were moving, or the sodium was moving in.
Simultaneously, repolarization of the first segment of membrane begins with the opening of voltage-gated potassium channels and the inactivation of the sodium channels.
So one thing I didn't mention, when that membrane thing becomes positive, what happened to the sodium channel? It closed, right? So it's just open for, a couple milliseconds, and then when it detects that it's positive, it's closed. So that now the only thing that's happening is the potassium's leaving.
The graded potential is unable to reach threshold and generate another action potential in the section of the membrane that is refractory. The nervous impulse is conducted along the membrane to the next segment as the graded potential there reaches threshold and self-propagates the action potential again. This cycle, or wave of depolarizations, continues until the impulse reaches the axon terminal.
All right. All right. So the first thing for y'all to do is correct a paragraph about all this business. So give you a few minutes on your own and then let you work in your group. All right, so let's make sure y'all got them. Is there anything wrong in the first sentence? Should say dendrite, not axon terminal. What about the second sentence? Should be positive ions. If you changed it to sodium ions, that'd be right too. Or you could say positive. What about the next sentence? If the movement of ions into the cell changes the membrane potential enough, action potential is generated. That one's good. Voltage-gated channels in the dendrite and the axon then open and allow Na plus ions to flow in. That one's good. This triggers voltage-gated Na channels in adjacent regions of the axon to open as well. That's good. And then dendrite and the axon, voltage-gated potassium channels open and allow potassium ions to flow into the cell. Out of the cell. So this is our repolarization starting. As soon as that wave of depolarization passes, that triggers the potassium channels to open. So positive ions can leave. And then once the membrane gets back to minus 70, voltage-gated potassium channels close again. That's right. And potassium pump pumps sodium ions into the cell and potassium ions into the cell. Sodium's pumped out. Exactly. All right. All right, so before we do our little activity thing, this is in your book somewhere, and it's nice because it shows you what's happening to the voltage or like the membrane potential, and it's color-coded, so it kind of tells you what channels are open or closed at that point, which is kind of helpful. So you know that, at the beginning, before you get to threshold or before anything happens, the sodium channels are closed, potassium channels are closed, and then if you get a stimulus to threshold, then the sodium channels open, and then the potassium channels open in green here, in yellow, and then when you're back to orange, the K plus channels are open, and the sodium channels are resting or waiting. Anyways, so if you turn over your worksheet there, got your instructions for this crazy thing I made up. I haven't tried before. And I already noticed when I was going through this that we're missing something. Well, no, I have popcorn kernels. But I mean in terms of one of our channels is missing. So on there, if you write somewhere, blue pipe cleaner is a sodium-potassium pump. And I'll give you some blue pipe cleaners. And if you also notice, it was blank for the color of Play-Doh, because I didn't know what color of Play-Doh I was going to give your group. So you should have some on your table, so you can write down what color you ended up with there.
Nasty.
Cool. All right. So highly suggest if you aren't totally sure how that all works, I'm sure you can find some materials at home to do this.
I have a picture of it, so I'll do it mentally.
No, I want you to do it for real. Get some Play-Doh or draw pictures, something. And you can just you can put all this stuff back up here.